be master of ceremonies for this final session. So I'd like to, in actual fact, there's a slight change in program running order here. So we were due to finish with Vivek Gupta from St. John's University on advancing oral modified delivery for rare diseases via lipid-based formulations. Vivek sends his apologies. He's a little bit unwell right now, so he's probably going to be joining us virtually tomorrow. So we took the liberty in this conflict to bring Dr. Ryan Pelis from Novartis over he was originally speaking tomorrow afternoon on the topic of cellular model of the oral lymphatic absorption pathway. So we're going to obviously switch gears a little bit here, and Ryan's going to be finishing up the day with us here. There'll be a little bit of time at the end for Q&A, and we've just got a couple of final announcements to make after Ryan's talk and before we break for the drinks reception. So please join me in welcoming Ryan as our final speaker of the day.
Yell at me if I don't.
You're good.
Thank you, everybody. I know everybody's tired. It's getting late. In full disclosure, I'm a biologist, so hopefully I don't bore you. Feel free to go ahead and lock the door. Keep people here. And if eyes do get droopy, I used to be a professor, so I can certainly easily call you out. That's not a problem. So yeah, I was at Novartis before being a professor for about 8 years. I was in East Hanover, New Jersey, and more recently, for the past 6 years, I've been in Cambridge, always in DMPK. And lately, Novartis has been making a lot of drugs with these weird PK profiles, pharmacokinetic profiles. Late Tmax, very long half-lives, very extended PK profiles. The question keeps coming up, is this molecule being developed potentially a candidate for taking the oral lymphatic absorption route following oral administration? We have other cases where we may want to target that pathway through medicinal chemistry or by co-administering excipients that will stimulate the oral lymphatic absorption pathway. So that's what I'll talk about today, trying to develop an in vitro cellular-based assay to investigate or probe this particular pathway. Really, right now, the gold standard for understanding oral lymphatics is lymph duct cannulated rat model, which is a very challenging surgery, not very successful. The success rate's only about, I think it's around 50%, and it's highly variable. So a lot of times and very expensive. So we'll have studies done, and they're really inconclusive a lot of the time. So can we build an in vitro model to study this pathway to try to prioritize our later downstream animal models? And that's not working. Can you hit the forward button? I didn't even see the pointer work. Is it working now? There it goes. So what we want to try to do is try to develop a cellular-based model where we can look at the fraction. Let's go back forward. This one, there, okay. Pointer. So following oral administration, what fraction of the oral dose goes the traditional hepatic portal route versus this alternative lymphatic route? So that's our goal. And this just shows a pharmacokinetic profile of vitamin D3. Here on the y-axis, the plasma concentration of vitamin D3 over time. These are lymph cannulated animals. So in these animals, what we're looking at is absorption into the traditional hepatic portal vein pathway. So you can see relatively low exposure compared to IV administration. And this is exposure in animals via both the lymphatic and the portal routes. So for vitamin D3, about 75% of its oral absorption occurs via the oral lymphatics or the mesentery lymphatics and ends up circulating in the blood circulation. And I'll get to some of the physiology behind that. So here's the physiology of the lymphatic system. There are multiple organs involved, and the intestine is one of them, and that's our focus for today. There's a lot of immune organs. It's very important for homeostasis of interstitial fluids. So fluid homeostasis, very important for dietary fat absorption, immune response, and the lymphatics, and this is important for this talk, compared to the blood circulatory system, it's highly permeable vasculature. Okay? And that's very important for its physiological function in terms of preventing, or maintaining fluid homeostasis, preventing edema to absorb interstitial fluid. But it's also very important in terms of the GI tract. So this is just a schematic of the intestinal lymphatics. So here are the villi, the crypts, and the pre-lymphatic channels are the vasculature, the lymphatic vasculature that we're interested in, highly permeable, and it is in the mesentery just below the anocytes of the intestinal epithelium. Now, following fatty meal, dietary fat ingestion, there's lipolysis that occurs in the intestinal lumen to form fatty acids and monoglycerides. Those fatty acids and monoglycerides are absorbed into the intestinal anocyte, and they're repackaged into triglycerides and then packaged into lipoproteins. And the lipoproteins from there are secreted across the basolateral membrane of the anocyte into the mesenteric compartment, or the interstitium, and they can go one of two routes. They can take the hepatic portal pathway, or they can take the lymphatic pathway. And there's various types of lipoproteins, very low density lipoproteins, high density lipoproteins, and chylomicrons, for example. Chylomicrons are the lipoprotein that tend to take the lymphatic pathway. Because they're so large, they're size excluded from entering into the blood vasculature and the hepatic portal system. So they tend to go the lymphatic route for distribution to the rest of the tissues, and then you can use the triglycerides and cholesterol. So because of this pathway and the implication of dietary lipids, with drugs that are absorbed via this pathway, such as this one is navitoclax, I think, we tend to get a positive food effect, okay? Especially with a fatty meal. They tend to stimulate the lymphatic absorption pathway, and drugs that go this pathway tend to get a boost in absorption. So a little bit more about lipoproteins and chylomicrons, which take this lymphatic route of oral absorption. They are the largest and least dense of the lipoproteins, so they're very large, low density. This is important for this talk. Another biomarker is that they're very rich in triglycerides compared to the other lipoproteins. And a biomarker of chylomicrons is ApoB48. So ApoB48, if you see that with a lipoprotein, it indicates it's a chylomicron, whereas VLDL and lower density lipoproteins tend to have ApoB100. I won't highlight this pathway too much, but there are actually two pathways. The glycerol-3 phosphate pathway, again, we have lipolysis that occurs with dietary lipids in the intestinal lumen to form fatty acids and monoglyceride. They're absorbed across the apical membrane in the intestinal anocyte, and they can go two pathways. This glycerol-3 phosphate pathway, where lipoproteins like VLDL tend to be absorbed in the hepatic portal system. And then there's this alternative pathway, this 2-monoglyceride reesterification pathway with a lot of enzymes that are involved. This is the one we're interested in that forms these chylomicrons containing lots of triglyceride and ApoB48 that when secreted across the intestinal anocyte end up in the mesenteric lymphatics. Now, there are quite a few molecules, drug-like molecules and drugs that take this pathway to different extents. It's not an all or nothing absorption pathway. Some are 90%, some are 5%. One of the, there are in silico models that are used to predict this. There's also an intralipid association assay. Basically, it's a chylomicron purified human. You can collect them from blood and mix them with a drug. What percent of drug incorporates into a chylomicron, what fraction. So there's a couple different assays that can be used, but there's no cellular-based assay. Triglyceride solubility is certainly one phys-chem property that may dictate whether a drug will take this pathway or not, but not necessarily. And there's also more recently been a lot of work looking at, and Novartis is getting into this, is excipients or formulations that may stimulate this pathway or allow a drug to take advantage of this pathway. Why do we care? Well, potentially we want to target the lymphatic system. For example, immune targeting, right? That may be one reason for targeting the lymphatic system following oral administration. Another example is we may want to bypass first-pass metabolism. If the drug doesn't go to the hepatic portal vein, there may be less first-pass metabolism due to hepatic, biliary excretion as well as metabolism. So we might want to bypass hepatic first pass. And this may be a good thing or a bad thing because lymphatic flow is much less than blood flow. You tend to get, as I mentioned already, these extended PK profiles. We get a late Tmax and extended PK. So those are just some examples of implications of going this route. So our goal is to develop an in vitro assay that captures or recapitulates the contribution of both pathways, the lymphatic and the portal pathway, to oral absorption. And some work had already been done with CACO2. This is kind of a gold standard model. And I'll present data on CACO2. This is kind of where we started. But we branched out and actually started working with primary intestinal tissue-derived organoids from duodenum. We also have ileum from human as well as rodent. So we have mouse intestinal organoids. So we examined both cell types for establishing this particular assay. So a little bit about intestinal organoids. They're three-dimensional tissue cultures derived from stem cells in the crypt. So we can take tissues, biopsies from human intestine. Depending, it's a lot easier to get it from the duodenum or the colon than the jejunum, but we can obtain jejunum. So you can just dissect the tissue, and you can take that tissue, and these are what I'll talk about is the tissue-derived intestinal organoids. They're multicellular. They contain all of the cell types, the epithelial cells, intestinal enterocytes, enteroendocrine cells, goblet cells, and more importantly, stem cells. We can break those organoids up, plate them on a transwell plate, and grow them to confluence, just like a CACO system. And then we can switch the media over and differentiate them. So they go from a stem cell population to a multicellular intestinal primary epithelium. Goblet cells, enteroendocrine cells, intestinal enterocytes. Unlike a CACO cell, which is a immortalized cancer cell line that derived from colon. So this is a tissue-derived organoid. You can actually see the villi here. We can break these up. We can put them in culture on a transwell, 96-well to a 6-well plate. And here's your epithelial monolayer that contains about 20% goblet cells, contains enteroendocrine cells, contains all of the cell populations. And we can actually grow them in either a symmetrical conditions with media on both sides or an air-liquid interface. So we can remove the media from the apical side, as you kind of have in the GI tract, and that will cause these cells to secrete mucus. CACO don't do this, okay? They secrete mucus So another interest we have with these cells is not only the lymphatic, but permeation enhancers, screening those, using them for predicting fraction absorbed and fraction escaping gut metabolism. So those are the areas we're kind of working in with these particular organoid models. Okay? And having that mucus there with formulations that are pretty stringent on the cells, Keiko fall apart, MDCK cells fall apart, but having the mucus there is protective and more physiological-like. So we can seed them on transwells, grow them out. And again, we're interested in this 2-mg pathway. There's a bunch of enzymes involved that convert monoglyceride fatty acids to triglycerides and then incorporate into a chylomicron that contain ApoB48. Experimental setup. Just move along here. We have our Keiko, we have our intestinal organoids from human and mouse. What we can do is plate them, differentiate them, and then we can actually treat them with dietary lipid formulations. Here's our control, taurocholate and phosphatidylcholine myceles, TCPC plus fatty acid, TCPC plus fatty acid plus this 2-monoline, which stimulates that 2-mg reesterification pathway that is involved in the formation of chylomicrons that are absorbed via this lymphatic route. These are the formulations. We can add them to the apical side for 2 to 4 hours. We treat these cells. We can remove that. So this is our pulse. We're pulsing these cells, telling them to, synthesize lipoprotein and secrete lipoprotein. So we pulse them, we can remove it, and we can look at biochemical profile, and we can look at permeability assessment across the epithelium of various drugs, some which are known to take this pathway, and maybe some that are not, and look at the contribution of the lymphatic and incorporation into these chylomicrons. So that's what we did. I won't go into this because of time, but we looked at markers of epithelial integrity, and for the most part, they do retain good integrity, the epithelium. They still form a nice barrier. Here are our formulations. Again, we can add them to the apical side for 4 hours. And what we can do is, after that, remove this, and we can do a time course experiment or a collection experiment where we remove the basolateral media, the secreta, and we can do density gradient ultracentrifugation of that basolateral media. And we can isolate out the different lipoprotein fractions. Again, chylomicrons are the largest, least dense. So when we do density gradient ultracentrifugation, those chylomicrons end up in this top 5 fractions, followed by VLDL and followed by the bulk media. So we can look at recovery of different biomarkers, and we can look at recovery of drug in the different fractions, in the chylomicron-rich fraction, the VLDL fraction versus the bulk. This is just looking at some of the biomarkers. This is apolipoprotein 100, B100. This is the VLDL marker. This is 48, a chylomicron marker. You can see that there's a lot of apoprotein B in Keiko, much less in the intestinal organoids. And with the intestinal organoids, they mainly have ApoB48 secretion into the basolateral compartment, whereas Keiko have mostly ApoB100. We can do ELISAs. So here's our control, taurocholate, phosphatidylcholine, and then we can add the fatty acid and then the 2-monoline to try to stimulate this pathway. And you can see in all 3 cell lines, we get this stimulation or boost in lipoprotein secretion, especially in these first 5 fractions, which is our chylomicron-rich fraction. So we can get this boost, and we can look at the profile of secretion. Now you can see this scale. This is a Keiko. This are the intestinal organoids. You can see, just as represented in the Western blot, there's a lot of ApoB secretion by Keiko, okay? But the ApoB that is secreted is mostly ApoB100, the solid, whereas the primaries secrete mostly ApoB48, which is our chylomicron marker. And Keiko, human duodenum, mouse lamb, This is just looking at the direction of secretion. Does it go apical to the apical compartment where it shouldn't go, or does it go basal into the mesentery? And blue means that mainly it's secreted in the basolateral direction, whereas CACO go in both directions. It's not quite sure which way to secrete the lipoprotein fraction. We looked at triglycerides, again, with chylomicrons having the most triglyceride, or being the most triglyceride rich. Here's CACO. You can see these scales are the same. So there's a lot of triglyceride or more triglyceride secretion by the primaries. And that's the same thing shown here. This is just the mean of that data. And we can actually look at the triglyceride load per particle, and that's shown here. And so, again, these scales are different. See, this goes up to 2, this goes up to 20. So per lipoprotein, for the primaries, there's a lot more triglyceride per lipoprotein. So that's more representative of a chylomicron. And we also looked at lipoprotein size with DLS. These are purified human chylomicrons. They're about 100 nanometers. And you can see they're a lot smaller and similar between the different cell types, although we do get this change in size when we pulse with the fatty acid and monoglyceride, which we would expect when we're stimulating this pathway. So it goes from the blue to the green. We looked at enzyme expression. Again, these are the enzymes involved in the 2-mg pathway to synthesize chylomicrons and package ApoB48 into it. CACO2, when we stimulate with these dietary lipid formulations, tend to downregulate these pathways, whereas at least the MGAT and DGAT pathways are upregulated in our primary intestinal epithelial cells. And then finally, in the next, I'll keep on time, I think, in the next 2 minutes, again, we can pulse with our dietary lipids, our control fatty acid, and then the formulation that stimulates this 2-mg pathway and chylomicron synthesis and secretion in CACO, as well as the primary intestinal epithelial cells. We can remove that, and then we can add drug. So now, after we've treated for 4 hours, we know we're pulsing them. They're going to synthesize these lipoproteins, and they're going to secrete them. So we remove our dietary lipid formation. Then we can add, on top of that, drugs that are known to take this pathway. And the two we chose were navitoclax and halofantrin. So I'll show you permeability data for those. So we can get permeability. We still collect the basolateral media. We can do fractionation, and we can look at drug in each fraction. Okay, so we can get complete recovery of how much we dosed and how much made it across, and in each fraction. 1 through 5 being chylomicron rich, 6 through 10 being VLDL, and then we have the bulk. So this is the data for halofantrin, and I'm just going to move forward here. Again, we can look at each of the fractions where the drug falls out. Then this is the chylomicron rich fraction, and we can do it under control conditions and when we stimulate this 2-mg pathway. And what we did was we did the pulse with the dietary lipid formulation, and then the chase, where we add our halofantrin in this case and look at its permeation across the epithelium. And we can collect for 2 hours or 4 hours, and then this is the combination of a 6-hour period of permeability assessment. Here's CACO2, here's our control condition, here's with our dietary lipid formulation that stimulates 2-mg pathway. So we get this like a nice positive dietary lipid food effect. And here is what we have, the 6-hour period with our primary intestinal epithelial cells. So what we've shown is around 20% takes this pathway in vitro, and it we kind of have a good IVIVC with what we see in vivo with the rat model. Around 20% of halofantrin orally administered takes the lymphatic route. And we did the same thing with navitoclax, and that data is shown here. Here's our CACO model, here's our intestinal epithelial model, and here's our 6 hours. We have a control, and we get this nice boost in permeability when we stimulate this 2-mg pathway. Okay? Even without stimulation, it's still taking the lipoprotein. It's still going via lipoproteins, but we can just get this nice boost when we prime the cells. So to keep on time, to conclude, we established a method using dietary lipid components to prime both CACO2 and intestinal organoids to synthesize and secrete lipoproteins. Engagement of the 2-mg pathway leads to lipoprotein secretion from both. The lipoprotein secreted from the primary intestinal epithelials seem to be more representative of a chylomicron lymphatic absorption pathway compared to the CACO, although we get similar results when we do permeability assessment with both. So these show, promise that this cellular model will work. This is a lot of work that went into this. A PhD student worked in our lab for a year, and then another year on top of it, starting with CACO and then going into the intestinal organoids. And he just finished and graduated from Northeastern. So that is Ian Smith. And so this was done in collaboration with Rebecca Carey at Northeastern University. Manuel Sanchez Felix, many of you may know, he's gone on to Halozyme, but he was at Novartis for a while and really worked with me to kind of set up this system and all of the collaborations that went into it. We get our organoids from Boston Children's Hospital in association with David Brault and Ramnik Xavier from Broad Institute, Harvard, and there's a bunch of people at Novartis that contributed. I didn't show any of the in vivo work, but we have a lot of in vivo data with the lymph duct cannulated model as well. So thank you.
Thank you very much, Ryan. All right. Any questions for Ryan? I know we've sort of switched gears and adjusted the programming slightly, but yes, Diffie.
Hi, Ryan. I really enjoyed your talk. It was a good departure from all the drug product conversation all day. Just a general question. Are there any molecular descriptors that you track? And if you see something, do you right away trigger this intestinal organoid assay, or it is done more retrospectively? When you see preclinical PK data that is, as you said, funky, then you go back and try to explain it with this in vitro assay. Just wanted to know about your workflow.
Yeah, I kind of touched on it when the first slide is this has come about because we see this weird PK profile. So it's retrospective, and that's why they've asked us to develop this model. We haven't even implemented it yet. The question is whether we're going to implement it internally or whether it's low throughput right now. And, the number of requests, we're still trying to feel whether, it makes sense to develop internally or just kind of outsource it to Rebecca's lab at this time, because she's still working on this model and we've collaborated. We're just about to publish a paper on it. But right now it's retrospective, but we have a lot of cases where we see this, and tends to be like high log P, the triglyceride solubility, long chain, those kind of like trigger our thoughts into this area. Yeah. But now we, Rebecca's interested in this as well, is, what formulations trigger this, right? What formula, because a lot of these lipid-based formulations were probably stimulating this pathway. We now have a system to test that. Yeah. Yep. Yeah, protein degrader, that's kind of why we're studying it, that as well as the permeation enhancers. Some of our protein degraders are just, the larger ones have terrible bioavailability.
All right, thank you for the talk. I had two questions. Is there a specific reason that you're using tissue-derived organoids instead of stem cell-derived organoids? And can you comment on kind of the reproducibility from like where you harvest and from the animals or humans, and the potential use of like stem cells where you could have control over monogenicity, could potentially even do knock-in and knock-out experiments with these organoids?
Opportunistic. We haven't tested the iPSCs. We haven't tested them yet. It would obviously be probably easier to work with because of availability. We get these. Novartis will have a problem getting them from Boston Children's, so that's why I've been collaborating on this with Northeastern. for other reasons as well, just the, you know, the intellectual. But we've also explored Altus Biosystems, so their platform is pretty good, and it's built on the same platform as where the tissue-derived. But we haven't expanded out into, you know, the true stem cell to see whether we could reproduce. I mean, the Keiko are doing something similar, but they're very different than what you'd see physiologically in vivo. But at least for our purposes, if we want to just look at transport, they seem to work perfectly fine. We have a small data set, right? We have to still build this up with negative controls and all of that to see how well the IVIBC works. So, yeah, this is still work in progress, but it's just so low throughput, and graduate students are great for this. Yeah. And reproducibility, we're kind of exploring that with the Altus Biosystems because they're commercially available, and they have 4 donors. And so at least for those 4 donors we could. And again, we can't really get them from Boston Children's. We'd have to do that through Rebecca. And she's doing other work with them to look at reproduce. She's really interested, and she can get a ton of different donors to look at that. And we just discussed that the other day.
All right. Thank you very much, Ryan. Wonderful talk.
Oh, yeah. Again, thank you, everyone. Thank you, presenters, for great content and state-of-art knowledge that you shared with us. Tomorrow, obviously,